I'm Annika Viswesh, a rising junior at Palo Alto High School, California. This is my project, Cancer Prediction Using DNA Methylation Profiles. DNA methylation is one type of epigenetic marker. Epigenetic markers influence the activity of the genes without altering the DNA sequence and help the cells carry out their normal functions. Methylation is an important component in numerous cellular processes. Changes in regular DNA methylation patterns can cause problems in regulating gene expression like growth signaling and apoptosis. A reduction or increase in DNA methylation can be seen in cancerous cells. DNA hypomethylation or hypermethylation can cause some genes to mutate to oncogenes, and it can also silence tumor suppressor genes, which in turn can cause cancer. I chose to study DNA methylation because research shows that DNA methylation patterns change early in the onset of some diseases. DNA methylation profiling can be done non-invasively, quickly, and cost-effectively compared to methods like biopsies. If we can determine cancer using changes in DNA methylation profiles, then we can use it in early cancer detection. Early cancer detection can improve the patient outcome significantly. The goal of my research was to create a computational model using machine learning in order to predict a cancer type based on DNA methylation profiles. Machine learning is the ability for a machine to learn patterns in data. Some of the steps involved are data preparation, building a computational model, and improving it. I used a freely available DNA methylation dataset from the Cancer Genome Atlas, TCGA. I chose 6 different cancer types based on the 6 different cell tissue types these cancers originate in. These are carcinoma, CNS, sarcoma, melanoma, adenocarcinoma, and leukemia. The number of records for each cancer type ranged from 140 records in acute myeloid leukemia to 536 records for brain lower-grade glioma. Here's a sample of a record from a TCGA dataset. Each column value indicates the degree of methylation, which ranges from 0 to 1. When I downloaded the data, I also added a column to indicate the cancer type. The cancer data had a lot of challenges. The records for each cancer varied from 140 to 536. This typically leads to underfitting problems. Another challenge with the data was the dimensionality problem. It had very less number of rows compared with very large number of columns. This typically leads to overfitting problems. Another challenge was a lot of missing values, which can cause errors in some algorithms like decision trees. To solve the data imbalance problem, I randomly undersampled and oversampled data and brought all cancer types to 200 per cancer type. To solve the missing values problem, I chose to impute the data with mean values. To solve the dimensionality problem, I used principal component analysis, or PCA, which is a type of dimensionality reduction technique. I experimented with a range of reduced dimensions to ensure that most of the variance was captured and reduced in the data to 1,200 principal components, which captured all the variances in the original dataset. The table shows the data after I wrangled it. Each cancer has 200 records and 1,200 columns. Since I had created the labels for my data, i.e., the cancer type, I decided to use supervised machine learning algorithms. I used logistic regression, support vector machines, Gaussian naive Bayes, decision tree, and random forest algorithms. I split my data into a test and train set. I also used tenfold cross-validation while training the model. I trained the model by tuning the hyperparameters for various algorithms. On computational models that were produced, I ran test data and checked my results. I repeated building several models by tuning different hyperparameters until the results met the metrics criteria. So here's how I evaluate my metrics. I use 3 evaluation metrics to evaluate my model: precision, recall, and F1 score. Precision is the ability of a classifier to not label a negative sample as positive. Recall is the ability of a classifier to find all positive samples. F1 score is the harmonic mean of recall and precision. Here's a plot of precision for different algorithms and different types of cancer. A high % of precision indicates that the algorithm is performing well. Here's a plot of the recall for different algorithms and different types of cancers. A high % of the recall indicates that the algorithm is performing well. Here are the results of the F1 score for different models and different types of cancer. A higher F1 score indicates that the algorithm is performing well. Gaussian naive Bayes generally performed poorly. Its precision and recall was typically low in the 40%, and its overall F1 score of 38.6%. Its poor prediction may be attributed to the algorithm's inherent assumption that all features are conditionally independent, which might have not been true for the methylation data. Decision tree performed well, with precision and recall in the 85% and overall F1 score of 87.4%. Random forest performed better than decision tree with a precision and recall of 93% and an F1 score of 93.6%. Logistic regression and support vector machines performed the best, producing precision recall and F1 score of 99.5%. PCA turned out to be a good technique for dimensionality reduction. Here are the conclusions. The results show that it is possible to identify cancer types based on DNA methylation profiles. The best machine learning algorithms for this purpose are logistic regression and SVM. The most important conclusion is that DNA methylation pattern changes occur early and can be done non-invasively, quickly, and cost-effectively. So this ML-based approach can be used in early cancer detection. Although my study utilizes only data for 6 cancer types, this ML-based study can be extended to incorporate more cancers in the future. Here are a few of my selected references. Thank you.